of the methods, okay, which you can explore and how it should go, and the flow chart of your experiments. Okay, means when you design your experiment, what should be your flow chart. Secondly, then how to execute these experiments, and further some case studies where we have done the real experimentation and its significance and output. So this will be 3-stage learning, okay, and other simulations and all that we will try to do in the smart class maybe tomorrow or day after tomorrow, okay, because we have still 4 days left, so we have enough time to do that. Okay, so these are few examples. If you see, this is a flow chart, okay. If you want to do protein structure prediction, I told you, and yesterday also you have learned, you need these methods to be in place. So when you design a paper, when you design any type of research based on your learnings or in this workshop or for your research, you need to see these methods first, experimental methods and computational methods. So we are more dealing along with computational methods when we talk about the structure biology, but along with that your computational methods are totally dependent on your experimental methods. So if you do only computational, nobody will appreciate. If you do only experimental, the impact will not come. Okay, so you need to have the balance. So like X-ray crystallography you have seen yesterday, so I'm not going to give all the details there, but pen down these methods. If you want to do combination of these methods, then you will be able to get the information. Along with that, you have to see the accuracy chart. If you see the accuracy chart from here to here, okay, so this is pure in silico. Most of the MSc students and basic research who are doing in bioinformatics, they do Ebrito modeling, and in silico modeling they try to see structures, they try to understand how the structure is coming up, they do simulations, they do modeling, okay, and this is very common. Every time we can see in the work. And this is simple also, because you take 1 or 2 molecules and you do docking, simulation, and you get the results. But coming to this part, Cryo-EM, NMR, X-ray, it requires lot of more time, lot of more quality, and whatever techniques explained to you, study, and especially the accuracy. So the accuracy is coming from all these upside methods. So you need to make a flow chart when you plan your research. You have to make that in silico methods combined with your wet lab methods. You get a better outcome of your research project. Okay. Now, when we talk about experimental methods, again the facilities are not available everywhere. Okay. So you have to have the idea that okay, if you have your samples ready today, okay, then you need to plan it in advance. So if your samples are ready today, you cannot book immediate Cryo-EM or NMR or X-ray. So which is the major problem? So in tutorial you should also learn that preparing the samples in advance, having samples ready in advance, and calculation about when the samples will be ready in case you want to send sample to somewhere else, like in Bangalore or some other places. You need to have your samples ready, book the slots accordingly. It is always a mismatch. Most of the time, because we don't discuss this thing in the tutorial, we learn technique, we don't learn how to adopt the technique. So how to adopt the technique, like if you are willing to send the samples today, you should have the samples ready and the booking has been done. It is not that as sample ready hai and you will get the booking tomorrow and the sample will reach tomorrow and it will be processed, you will get the results. No, your sample will be wasted. And many researchers who are participating, they know few of the analysis we need to do at IIC Bangalore or IIT Kanpur or IIT Delhi, anywhere. You want to prepare the samples in advance, make a time flow chart. I have not shown it here, but time flow chart is very important. If you want, I can give you the template. That is very important. So time flow chart is very important to know when to send the sample for Cryo-EM, NMR and X-ray. Okay, and you cannot have always positive results. You should also have a backup plan that in case your samples are not enough, they say the quality is not good. Recently we experienced some restrictions in the samples quality. Okay, so that is my own experience. Maybe others have their own experience. Then they will ask you, they will give you 48 hours. Okay, reply whether you want to process this sample or you want to withdraw the sample. Then you have to have. That's why timesheet is very important. So we have to reply them. Okay, proceed with this sample. The risk is with us because the sample already sent. The payment also to be made by us. So the method which you are using and the preparation of the sample is very important. So probably yesterday in tutorial you have learned how to prepare the sample. So I'm not focusing on preparing the sample part. Okay, going to the next one is the experimental protein structure determination. Okay, so if you see these three techniques, okay, so most people they have not followed all the techniques in one paper, but maybe two techniques or one technique if you follow, the accuracy and the impact will be always at a limited place. So if you follow electron microscopy like imaging technology, okay, and you just see what is happening. I will show you some electron microscopy image also. Okay, so you can get that, like if you are looking for a protein drug interaction or protein modeling or protein crystallization, whatever you are looking, electron microscopy is an imaging technology. So you can get only the images and you can understand, make legends and understand that. When you go to NMR, according to that it is fairly accurate. It is not fully accurate, fairly accurate. Means NMR based data, sometimes they may also mimic to other structure also. Okay, so they are telling fairly accurate, but MNR is also very good. Okay, then no need for crystals. This is the bonus point for you. Okay, like yesterday we told making crystals. Okay, protein crystal is a big challenge. So the major problem of crystallization and the proteins which are not crystallized properly is sorted out. Then you have limited to very small proteins. This is another thing. This is the limitation. Okay, so limitation is only small proteins. Then time consuming. So time I already briefed you. So these are the limitations and some good points. So every technique has some expensive, some limitations and some strength. So you have to, that's why I tell you time sheet, plus and minus, and then plan which technique you want to use. Most of the time, what we do in our labs, what we do is that we get a problem, we start with objective, we keep on going. We never see back and we don't make a backup plan. We never see also the future of it. We go on 3 years, if PhD 3-4 years, if MSc 1 year, 1.5 years. We go back, and if we go back with the plan which we made and reframe the plan in between, if there is some negative results or something is not coming, then I think it will be a better thing which you should learn. So my suggestion in all these techniques will be that when you are taking these techniques in your research, you should have a backup plan and also go back and see what you have decided. I have never seen students making or we researchers making a TOC. Okay, so I insist always, and my suggestion to you in this tutorial is make TOC, table of content, what you are going to do. And if you are not able to make a TOC, it means you don't have any idea. So don't do anything without any idea, without any confidence. Try to build up confidence, because these are very sophisticated techniques, time consuming, and if you see the X-ray crystallography, it is most accurate. Okay, in vitro needs crystals, time consuming and expensive. Okay, so when you talk about expensive, I don't know the current rate of IIC Bangalore of this X-ray crystallography. Nivas, can you tell what may be the cost right now? Maybe for the proteins, specific proteins. You can check it, okay, and we will discuss, because the cost is also very important. So you are sending sample is not good, okay, or sample is of not good quality, and you are sending for X-ray crystallography and you get a negative result, or you don't get a result proper, then it is a complete loss of resources, your own hard work, and also the time. So everything is compromised. So it is not always easy that you will get the positive results. It is not always good to look for positive result. Maybe you get a negative result also, but before that I suggest that you should see, make a TOC, make a plan of work, make a side means alternate plan of work. Also see what other techniques can help you. Okay, and if you are not sure that the crystals are not available or crystals will be difficult to have, then you can have NMR only, or you can have electron microscopy based on your own requirement and your literature search. The second part which is not written here is about literature search. Most of the time we fail in reading. Most of the time we all fail in reading. We go directly to the experiment without reading properly. So when you make a TOC, you will definitely read it. For reading, you have to collect the new papers, some older papers also. But if you try to adopt a technique which is published 10 years back, then you may have some compromised results, because the techniques is upgraded. Okay, so you need to see that whether you are following these techniques and how to manage these techniques in your research. That is important here. Now, computational methods, which I already covered, and these are the major techniques: template based modeling, homology modeling, threading, and template free modeling. Okay, where you have this knowledge base without use of known protein structure. So most of the time we do the template based modeling because we know the protein structure, but in case you are working with hypothetical proteins or you are working with some other protein structures, you can also use this template free modeling. Now homology modeling I don't want to brief because most of you use it, and you are doing tutorials in your MSc also, in your practical also, in your research also. But here I want to tell you is that homology modeling is the simplest way of modeling the proteins, and it will give you a sense of satisfaction that okay, I can handle the homology modeling techniques. Means it's a trial. Okay, it is not a foolproof plan, but it is a trial. Okay, so when you do homology modeling, you understand what is sequence similarity, how to do the sequence search, what sequence to select. Okay, and then all other specific tutorials which you learn during homology modeling. Then if there is no homology, means the modeling is not possible. So this is the limitation here, but the servers which you can use is the Modbase, Swiss model, and Modeler also. So these are the servers, you can go through these servers. They are very simple, and this is the simplest way of understanding the simulation and modeling. I am not talking about simulation right now, but molecular modeling. Okay. Then homology modeling, what will be the steps? Okay, these are the steps: template selection, sequence alignment, model building, backbone model building, loop modeling, side chain refinement, model refinement, and model evaluation. Okay, and the softwares which are to be used are given in the bracket. Okay, and if you see this 6th step, if you don't do a single step, your experiment is not complete. Okay, so many people try to not do this loop model. Okay, because this is some time-taking activity, and if you are handling so many structures all together or doing homology modeling of many other objectives, then it is a time-taking step. And also this model evaluation, most of the people they do because they know that after evaluation of the model they see the accuracy of the model. Side chain refinement is also challenging. Okay, so these are simple things you can try with 1 or 2 proteins which we can give you. Okay, in the smart classroom where you have this mouse facility, if you have mouse. Okay, but if you don't have mouse, then you can not do it accurately, but you can manage it. But the thing is which important is that 30% sequence identity fold into similar structure, that when you try to see that there is a alignment issues or there is side chain refinement issues or model evaluation is not proper, then again you go back to the template selection and redo, so that you are able to have that structure modeling of that particular protein well documented. Okay, and most of the time what is happening is that I have never seen any student coming up with that I am not able to do the model. Everybody is happy and successful. Okay, I don't know why, but most of the time we are happy and successful in that. There should be some analysis which is not written here. The model which you have built to be compared with other models which are built by other groups, published models. So none of people they do that. Okay, if you do a comparative analysis, you can note it down. A comparative analysis of the similar models, okay, built by other researcher is important. Okay, so if you do the comparative analysis, you can understand that okay, your model accuracy, your model structure, okay, and other parts which you want to do. Then remote homologs may this technique will not work, okay, because it has less than 30% of identity. Now if you see this one, I suggest you to apply this DBTL cycle in your research related to this bioinformatics, computational biology, structural biology. And if you see this picture which is not much clear, okay, but the high resolution picture I can show you, I can give you the paper. This is about the DBTL cycle, design, build, test, learn cycle. Okay, now when you talk about design, build, test, learn cycle, the cycle name itself suggests what has to be done. Okay, first design, then build, then test, and then learn. Okay, so this cycle we did published this work in 2018, and that time we tried for it for phytoremediation. Okay, that is the plant-based bioremediation through microbiome engineering. Okay, so we use the principles of microbiome engineering to understand how the plant will be effective for how they are giving that signals. Okay, what type of proteins they are releasing, how the signal transduction is happening, and how the plant is remediating. Okay, because we understand that plant itself is not doing. There is some signals, there are some assembly of microbes. So for that you need to see that. Now when you adopt this technique in drug discovery or in other fields where you are working, you can see that transcriptomic is the key of that. Okay, then when you have this transcriptome data available, okay, you have to see that how it is connected and why this type of proteins are analyzed and how many proteins are giving proper indication that they have certain roles. There may be some proteins which have not important roles. So if you are looking for a role in some of the drug binding, if you are looking for a role in some process, if you are looking for a role in some degradation, if you are looking for a role in any type of disease modulation, so you have to see that you should have the information about transcriptome data. Along with that, if you are able to do the network analysis, okay, network analysis means a specific protein network, okay, and if you are able to build up network analysis, which is possible only when you have complete transcriptome data and also the data of this metabolome. Okay, so when you have this proteome, metabolome and transcriptome data available, you can build up the network. Okay, and when you are able to show the network, you can try for seeing what are the key networks. Okay, like if you want to improve some of the capability of particular microbe or particular gene. Okay, so for that you need to understand how this gene is triggered. So here we have shown you that the gene which are triggered for improved phytoremediation can have some triggering through specific type of network. So what is that network? The network is not easy to build up. Okay. Recently there is a technology which is WGCNA technology, which has been, we are learning about it. So if you see that network technology, it will give you a consolidated cascade of networks. And this technology is gaining lot of importance. I will send you few papers recently published, and the expert who is expert in gene network analysis and all that, we are inviting him in the next GIAN program in June. Okay, so June he will be here, and the network analysis is a very important component. For one fungi, we are building a network since 8 months, and now we are successful in, I think last month we are successful, but not yet have identified crucial network responsible for the application which we are looking for. So still the work is not complete, I say, but at least network we have built up. Okay. In the same way, when you start one organism, I am telling. So if you work on multiple organisms, you are working on metabolome data, you are working on lot of pathogens, you are working on drug discovery aspect, you should have lot of information. So it is very challenging for you to decide on that. And if you are working on especially animal cell culture or other aspects, because I don't have much expertise on that, so that is other challenges which is not in your control. So you should have all this information, and you build the network analysis, then you go what are the key pathways, okay, and then do the pathway modeling, remodeling and analysis. Three things are there: pathway modeling, pathway remodeling and analysis. Okay, so if you want to understand that which pathway is giving proper characteristic for the function you are looking for, you analyze that pathway and then go ahead with your gene editing part. You know now these genes are having crucial roles. If we alter, we change, we edit, we get the function done. So this is all called design, build, test, learn cycle, DBTL cycle, and we have adopted this, few of this in some of our research, but not exactly all. And I will tell you the text part of it, I have already explained you. The design and build component validates the gene expression, the proteins and metabolites. The learn component analyzes the large data sets generated by the design component through systems analysis. So this cycle is a combinatory aspect of learning. This is a complete tutorial for you for your complete research analysis, and information gathered by design and build components can be employed for identification of different gene expression and candidate gene with phytoremediation, because we are looking for phytoremediation functions, and we were targeting that PGPMOs, that is a plant growth promoting microorganisms. So in the same way you can see, and how these can be contributing to different attributes. So this case study which I am telling you is about the attributes. Now, is it successful? The question will be, is it successful? So the successful examples are here. Okay, means you can have a designer microbe. If you are really targeting a specific type of, if you are having DBTL cycle in place for the function, you can have a designer microbe, or whatever designer means you want to have, or a clone, or whatever specific target you are looking for. So these are the successful examples, identified microbes and how they have been used for improved phytoremediation, means the capability improved already, and these are published literature. Okay, so if you see this, this is the host, this is the gene, this is the target. Okay, and this is the function. Okay, so people, they like, if you see help in cleaning of uranium, if you ask somebody uranium cleaning microbe, okay, so this is the host, this is the gene which was targeted using this approach, and this, you see the target organism, and this organism is capable of doing that functions. So this is very big achievement of many researchers, and then we have published this in Trends in Biotechnology, Engineering PGPMOs through Gene Editing and Systems Biology, a solution for phytoremediation. question mark. Question mark is still there. Okay, because if we are successful, whether we can do it large scale, okay, means phytoremediation of the large scale, means large area, and also the area which is really bigger in size. So what is the challenge? That is the thing. And I think when you talk about these aspects, definitely this paper is good read, and it's a tutorial paper actually. That's why I'm showing you. This is a tutorial paper, and everything I cannot write here, but in the next 2 pages, 3 pages, you will read all the terminologies, definitions, keywords, okay, and the techniques, about the techniques. So this paper I will share with you. Please remember the papers which I am telling to share, okay, because in case I forget, okay, making promise is fine, but forgetting promise is very common, okay. So please remember and take these papers because they are available in the lab, so I can share with you. And this we collaborated with US team, where this Royal was working, and Supratim and Sangeeta, they all three are very senior researchers, okay, and they are now in Mexico consortium. They have done antenna engineering now, okay. Antenna engineering is a different story, okay. So in cyanobacteria algae, they do antenna engineering to sense proper type of metabolites production. So that is another one. So they are in national lab and ML. And next piece, if you see this one, scope and application. This is important thing. If you are able to do these type of strategies planned, okay, I am not telling plan everything, plan 1-2 things, at least 1-2 things you plan. If you are having interest, DBTL cycle, if you apply in 4 years, 5 years, whatever time you devote in your PhD or postdoc or in your research career, which is unlimited, okay, up to retirement, definitely you can have more outlook towards its application. So you can see that current knowledge of system biology gene editing will establish the attribute, and then practical application is also very much, but the challenge is the organism's diversity, okay. There are so many organisms in soil. In the same way, when you talk about gut microbiota, like drug discovery is not possible without understanding the gut microbiota. So nowadays, all medicines which are targeted, they are also understanding the gut microbiota. Gut microbiota is the biggest achievement. Understanding of gut microbiota is biggest achievement. So if people are knowing about gut microbiota, and personalized medicine, which I was telling in the morning, is also connected with the gut microbiome. And in the same way, if you are able to see the diversity of organisms present in your gut, when you are having that particular drug, how many types of microbes are there, what are the functions, how they are active, how they are suppressed, how they are doing their job, okay. So this is the biggest challenge among us, all researchers. When you talk about drug discovery, also think about other aspects, okay, because drug discovery should be balanced with your own body's homeostasis, and also understanding the total concept of your body. Because whatever drug you are taking, I told you after trial it is coming, but trial is on limited people, okay. So what will happen, like in COVID you heard so many stories, okay? I say stories, I don't say the fact, okay. So when you see so many stories and you believe 1 or 2, or somebody believe all, okay, so this becomes a big problem for you to adopt it. So this is the field which needs further exploration, but as a tutorial you can read that paper and terminologies, try to adopt few of them and try to see how we go. Now this is a good tutorial slide, and I think again I will say that most of us, we don't use these softwares, okay, because they are good software. Some of you use it, some of you don't use it, okay, because some of you don't know about it. These are the software, they do not need any specific license, they are free to use. For functional category, like if you want to know the functional category, you can use COG, okay, and Megan, MegaRep, MGRAS people are using. Okay. Then taxonomic analysis. If you have interest only in the taxonomic analysis of a microbe or whatever you are working, you can use this domain analysis softwares, class, order, family, QUIM, other, and MLST. Okay. If you want to know the clustering, okay, like OUT, tree building, alpha diversity, beta diversity, taxonomic composition, rank abundance, okay, you use Galaxy, IMGM, Megan. And if you are interested in pathway analysis or pathway structure, pathway remodeling, pathway understanding, you can use this camera, Metalook, Unifrac. So many students, many researchers, many faculty, they are using these softwares, and these softwares are based on data. ऐसा नहीं कि software use करना है. You should have proper data, input data, then output data. Okay. The first thing is that it will not beat us. Okay. Second, you should have data. Otherwise, it will say where is data. So if you don't have data, these softwares you cannot use. With template data you can use, and each of analysis will give you a new angle to your understanding. New angle to your understanding. If you are working on pathogenic microbe, and if you want to do the clustering analysis, okay, like alpha diversity, beta diversity, you can understand the difference between pathogens, different pathogens, how they are specific to certain type of population. If you are working on some specific type of drug design, pathogen understanding is very important. How to design a separate proper drug without understanding its specificity. So immunogenicity, immunological tolerance, the pathogenicity, specific receptors, all that is through various type of understanding and techniques. And this is, I think I'll suggest you to use this software for your metagenomics data or general data, whatever you have, and try to learn it. See, these are some case studies, okay? Because it's a tutorial, I need to tell you some case studies, okay, on a specific microbes. So if you see, this is about, first I will say, tell you the background. Okay, so we were investigating the role of microbes in heavy metal degradation. Okay, so how this heavy metal resistance is coming, and how the microbes can be helpful for heavy metal degradation in the soil. So we isolated microbes from soil, and the soil we cannot isolate from everywhere. We have to go to the where the heavy metal is dumped. You cannot get heavy metal degrading microbe from anywhere. Okay, so you go to heavy metal dumping site, or where this is dumped, or nearby some all those things, and you collect microbes. So we did that, and after that we did the whole genome annotation, and then we did the REST software to see what is the multi-metal resistance potential. How these microbes, we understood that okay, these microbes are there. I am not telling you all the techniques which we used in wet lab, because it's a complete dry lab course. So those microbes we have selected based on their wet lab profile and other data which is available, which I will show you in the paper. After that we understand that okay, the genome of these two, because they are doing the job, we know from the wet lab. Okay, we got the idea that these two microbes are very good degrader, and they have a very good heavy metal degrading capability, and they can be used for bioremediation, bioaccumulation. Now, when you have these microbes, the question comes in mind of a person who is reading bioinformatics or structural biology: what are the proteins which are doing the job? How the genes are active to degrade those type of pollutants? So we see that we have these specific cobalt, zinc, cadmium resistance protein and mercury reductase, which are responsible for cobalt, zinc, cadmium resistance and mercury detoxification. So after that, it also contains, because when we did the analysis, these genes like SCS A, B, C and D genes were also important and responsible for copper sensitivity and binding, helps protect the microbial cells from copper stress. Why this microbe was not dying under extreme heavy metal stress? Because of these specific genes and proteins. So from dry lab From wet lab you confirm it is doing the job. From whole genome data you understand, okay, there are genes. Then what are the functions? So you have to do that. And now if you want to study a protein, if you want to crystallize that protein, that is further part. That is the innovation and high impact. So by the time you plan all these activities, it is already 4-5 years. So after that the work stops. So this work also stopped. Okay, so we could not achieve the protein structure and all those things. So that is the thing, we have to plan in advance all those things so that we can get the idea. Now, because it's a tutorial, again I am telling you, you should see that what microbe you are working, what specific problem you are working right now in your PhD or in your master's project, and try to see whether you can learn further on that. After you finish your master's here, you can learn further on that, or you can learn later based on your opportunities, whatever opportunities you get. Okay, so then I will show you the complete chart. Additionally, the genes of chromium means these two microbes, they found all the major genes which are responsible for heavy metal degradation. It means that these are the very good microbes, and we didn't engineer it because it is doing the job naturally. So when you talk about engineered microbes, this is something which is natural, and they were also reported with some metal specific transporters. We have not yet studied all the transporter genes. Okay, so we have not studied. So these are the transporters, and table 14 I am referring here. So this is the table 14, I think. Okay, and you can see these are the specific genes and heavy metal transporter proteins, and they are doing the job like this. So we were trying to see a story of understanding the microbes, how they degrade the metal, and also understand how the things can be related to other disciplines. So when you talk about any specific problem related to your research, this is a very good model which you can take up. These are the transport, this is the copper, and you can see here this is the, now similar thing when we talk about the drug discovery. In the morning I was telling about ARG, antibiotic resistance gene, AMR. Okay, there is, means I say it is a big problem, coming up problem, and every country is looking a solution, and India is one of them. AMR. If you get any type of infection, drug discovery is impossible because you don't have a drug, because antibiotic resistance is so much. Okay, and most of the time you see that antibiotic is not effective. Okay, and the doctors nowadays they are advising antibiotic sensitivity test. Then they will prescribe you the medicine. So why this drug discovery? This drug discovery because we should understand that also. Use and misuse of drugs. Use and misuse of antibiotics. When to use, when not to use. Most, I don't know how many times in a year you eat antibiotics, you take antibiotics. How many times in a year? In general, जब सर्दी खांसी होती है, whenever you get, how many times? You take antibiotics? अभी कोई बताएगा नहीं. People they take antibiotics, doctors they prescribe. We know here in BSU, and they prescribe the medicine and you take. Okay, जल्दी ठीक हो जाए, जल्दी ठीक होना. You will be all right in 4-5 days, 6 days. Even if you take antibiotic, if you don't take antibiotic, 4-5 days it will take. So when we talk about this drug discovery, we should always think about antibiotic use and misuse. So when I am talking about this heavy metal along with the ARG also, there are ARG, okay, antibiotic resistance genes which are found. If you see, there was a study in Ganga water, it was studied that many, and then some other water streams, okay, where this ARG was identified, and also people they noticed that the hospital waste which is thrown here and there, that is also creating lot of havoc for an ARG. So the medicines will be ineffective. Okay, most of the medicines which are used will be ineffective after some time. So what you will do? You will discover a new antibiotic. It is impossible. New antibiotic will take a lot of time. So you have to have the idea that this type of things are important, and this you can use in ARG also, in drug discovery as well. Now we did the in silico. This is also a new technique, okay, which is mostly people they are using these days in research. You can analyze the functional annotation of the key genes. So whatever pathogen microbe you are working, you can do this functional annotation. And I'm giving you example as a case study, but if you are working with pathogens, if you are working with a specific type of microbes, or if you are working with any type of research, you can do the functional annotation, and it will tell you the real story. And this type of whole genome annotation chart will come through RAST software. Okay, so you can know the what are the specific criteria, and then what are the how many types of genes are there, stress response, amino acid derivative, sulfur metabolism, carbohydrate. Means it is having a lot of data. Okay, so each data you need to analyze and you target. Okay, like in this program, fatty acid metabolism is the major. Okay, because that is also having very good role for diabetes and all these research. Okay, so therapeutic research and conventional research, you have to see the difference. Therapeutic research, when you talk about, you are very focused. Okay, I am working for this particular disease cure, like diabetes cure. Okay, and I need to pick this one. Hello. अभी मैं लेक्चर में हूँ, बात में बात करूँ। अभी मैं बिजी हूँ ना लेक्चर में, सॉरी। Okay, so this is the RAST generated through RAST, and it is easy, not tough one. Okay, it is all computer generated. Now, this I already I think told you about that. This is the copper tolerance. Okay, so in summary, this case study will give you this profile, that the wet lab exercise, MIC, AAS studies. Then you have the adsorption efficiency studies, and you got that, okay, this is can accumulate 79% and 85 and 71% of the lead. Okay, means they can absorb this much lead because of the capability, multi-metal resistance capability. And in silico studies, it all I already, and this we published in 2021 in Journal of Hazardous Materials, and lead bioaccumulation mediated Bacillus cereus BPS9 from industrial waste contaminated and their transporters. Okay, we are not able to complete the study of transporters. Okay, so this study is not yet completed, but we have used few of these microbes because we know this BPS9 and another study is there. So we know that, okay, this microbe is good. So we have used this microbe for Syncom, in which one of our postdoc were working. That is a synthetic community. Okay, and the synthetic community which I showed you in the morning. Okay, synthetic consortium. So we have used this one of these microbes because we know now that, okay, this microbe is good. Okay, and this is a comparative analysis where the techniques of in silico techniques were used. A comparative analysis of heavy metal bioaccumulation and functional gene annotation towards multiple metal resistance potential by this BPS20 Ochrabacter and BPS26. So BPS9, 26 and 20. These are isolated from the natural environments, and they have been proved to be a very good potential for biology. And they can have more. They have the genes also. I could not show you, but they have the genes for ARG also, antibiotic resistance genes. They can also degrade antibiotics, but we have not explored it yet. So we have the potential. These strengths are with us, okay, and hopefully some of will come. The next part of tutorial is strain designing. Okay, when you work on a specific type of drug development, you also try to see that, okay, can there will be any strain which is very good against any type of treatment means they can give good response in the terms of killing. Okay, so how to do that? So if you see, these are few techniques, please note it down, these are very good techniques you can apply in any research. The first is genome scale metabolic network construction. This is a good technique. The second is making the specific model and kinetic model of FBA between 2 or 3 compounds together which are targeting, and then strain engineering. So you develop gene knockouts, then you try to see specific upregulation and down regulation, and you can have a strain, but if the strain growth profile, growth kinetics, everything is not studied well, so your strain will not be able to have the metabolic yield, proper metabolic yield. So this is about that you use these techniques, and for that you need to understand some concept of kinetics. Also you can try to see the pathways, okay, specific pathways, how they can be used. So going further on this approach is more explained here in this slide. This also we have published recently. Okay, this is multi and integrated omics approach for understanding the cellular metabolism for strain engineering. Cellular metabolism, okay, whatever we are targeting, diseases or what type of pathogen, whatever we are targeting, we are not having any idea about this. We don't go beyond the reading, normal reading. So why not we see the multi-omics data? Again, DBTL cycle is there, you can see that. Okay. So when you talk about this integrated multi-omics, why you don't apply with the research with the microbe, with the specific cellular metabolism which you are working, this type of approach? So this is also good, and because of machine learning tools, because of AI tools, you can have the optimization of process. Like cellular process, you can optimize, plan the optimization step. Recently, there are some specific tools, I'm not covering those tools here, that is the machine learning tools. For that you have to learn the MATLAB. Okay, so that is AI/ML tools. Okay, so you can use those AI/ML tools and to optimize the process, and if you are able to optimize it in a better way, it is not always true that AI/ML will give you enhanced or a better outlook. It may give below also. Okay, so you have to see that how AI/ML to be used. Okay, it will not always give you the better one, but the compromised processes where the production is not proper, like if you are looking for an anti-diabetic compound, okay, or if you are looking for anti-cancerous molecule, anti-cancerous for your drug development, why not to use these tools for process design? That will give you a better outcome. One more thing here is not written, that is about using various type of metabolic engineering tools which I have written earlier. That also can give you a better strain. Okay, and if you are able to have a better strain for your product, for your metabolite, for your compound which you are looking for, definitely drug development will be a better one. Okay, so this is a key slide for drug discovery. If this researcher is using these techniques, most of the researchers they are using, okay, and the techniques are just here and there, but this slide gives you a complete profile that okay, if you use these techniques, you can have a better one, though it is not easy. Now, if you read this paper, this is a very new one, 2022, Metabolic Systems Biology and Multi-Omics of Cyanobacteria, because our group was working in that. So this tutorial, this review paper will give you the various techniques. Okay, if you read this paper and try to read, review and research both papers. Okay, because in research you get a one direction. This done, this done, this done, this done. Okay, and in review you see the techniques, you learn techniques. Tutorial, what is tutorial? If you are able to learn the techniques properly and adapt in the lab, in one go it will not happen. Second also not happen. But after 2-3 repeated experiments, trials, you will be successful. But how to know these techniques together? So you have to read these type of reviews. So I have covered this here, and this is also on this this sigma factor modulation. In pathogens also sigma factor a great role. Okay, so yesterday in the evening we were discussing with Professor Tong. He told that he is not covering the sigma factor right now, but there is great role of sigma factors, especially when you talk about the complex pathogens, when you talk about the complex molecules. If you talk about some biometabolites, sigma factor is very important because it controls the regulation, transcription regulation. And this tutorial paper, this is a review paper. Okay, there is review paper, opinion paper, forum paper. Okay, so if you see these review papers, they are all tutorial papers actually. So they teach you what is the technique, what is the state of art. So here also I have described all that thing, but you have to see that when you use the techniques, new techniques, okay, like strain engineering called Global Transcription Machinery Engineering, GTME. Have you heard about it? GTME. Okay, so this is a new platform. You can see the glossary here. I have clicked it. Okay, I will give you the paper. This is, I think it needs another 5-6 slides to complete this one. Then if the time permits, I will cover that also. But this GTME is actually based on CRISPR-Cas system, and many of the researchers. Hello. Lecture no. Urgent है कुछ? बैक कर दो तो फिर मैं कल का देखूँ. नहीं तो वो कर सकते हो ना. कहाँ? 10 मिनट के ब्रेक के बाद. Okay, so this is how and this is which is based on this CRISPR-Cas system, and I think we are still not thinking about in that direction, especially when we talk about the microbiome and therapeutics. We have not yet thought about it because there is a lot of things which you need to see and many diseases can be cured. And in the morning during the interaction with university PRO team, he told about the diabetes control and specific medicines for obesity control. Okay? Obesity is big problem. Okay, so there are some magic bullets coming up, okay, which will solve the obesity problem. How patle ho jao. So these are risky, but very safe. Okay, so how these medicines develop? How these very costly drugs? They are not easy for everybody to purchase that. Very costly drugs. Okay, but what they are doing, how they are doing, that is another thing. And then how it behaves with your body, how the body is behaving to that medicine, that is another thing. So because we talk on drug discovery, we talk on new techniques, we always discuss this. And these type of drugs, if you Google it, the sale of that drug which is launched recently, okay, I don't know, Pfizer or some other company, I could not remember, sorry, but that company, I think Monica might be having some idea because she is working on diabetes research, so she might be having idea. You know about that drug? You didn't read it. You people are busy actually, you don't read it, okay? So I will read on behalf of you and tell you. So this type of drug which is developed is good for the people who are 200 kilo, 300 kilo, okay, not for the person who are normal, okay? So there is misconception, okay? So then the surgery option is there. Then these things are, why these things are coming up? Because of specific research which is concentrated on one area, like they might be targeting a specific gene, okay, based on that they have blocked the receptor, whatever is there. and they are giving the drug. Okay, this drug you take, you will be all right, without surgery, without anything, without exercise. Everybody is happy. Okay, but the side effects, the real need of that type of drug, why the drug was not needed earlier, okay, why it is needed right now, that is also there. So these things are there, and for me the healthy body should have less drug, okay. So healthy body should have a normal routine, but drug discovery is important for the specific problems which are coming, because infections you are not bringing, they are coming, okay, and every year you are getting a new infection. And I think I shared with MSc student in the first semester how many new viruses are coming. No, how many? So many new viruses are coming. There was a science paper, I think 1 year back, that lakhs of cryptic viruses and their genomes they have studied from ocean water, and they are yet to arrive, and they are looking for a specific host, okay, and once they got our body they will be happy, okay, and then we transfer to each other, okay. So this is the thing which is very common, and drug discovery, vaccine discovery, it is all coming up because of those type of viruses, emerging viruses, okay. The viruses which name you only read in book, they are now coming up, okay. So this is that's why we have to think about ourselves also. Then this I already told you, okay, this I already told you. Now this is actually Tong has shared me these slides, few of the slides, and he told me that the main problem, misfolded state problem, how to see with this phosphoglycerate kinase protein. This is a protein they are targeting, and paper advanced. What is not printed with the help of the aspects can be covered, and okay, that the protein survivability and the complex survivability is also in that. So I think this paper you can download because it may be available, and I think it is here also. Okay, it is here, okay. So you see this protein misfolding involving entanglement provides a structural explanation of the origin of stressed or exponential refolding kinetics, okay. So this paper is a very good paper, and this is a new one, I think, okay. Recently it is coming up, and this is a good paper, and I think another one is there, okay. So just we will discuss it later. Let's have a 5 to 10 minutes break so that you can pause and then see other research areas to cover and probably this paper is not Tong's paper, another one is there, so I will share that paper as well. Okay, so we'll take up further 5 minutes, 7 minutes break because 1 hour is over, so take a pause or you want to continue? Pause? इसको नींद आ रही है, आप थोड़ा वो कर लो. Sleeping is also good. Life. We are life. So the people will listen to me. Okay, sleeping is good. How is it? Articles which gives you about enzymes which are used in various applications, that is synthetic biology and biocomputational approaches, and the author of this paper, the first author, is sitting here. Okay, he is the Mandeep. Okay, so he is right now in Hansraj College as assistant professor DU. So he wrote this paper and he has the copy, so you can get it. I also can give you the copy. And this is also a very good tutorial paper where we have listed many techniques. Okay, like if you see, and it is not with one enzyme which we have written here because we cannot cover all enzymes. So the enzymes which are used for therapeutic applications, what we have covered previously, they can also be studied. Okay, so the techniques are common. Techniques are almost common. Enzymes can be different. The techniques can be also used with those enzymes. So this is a recent article which is published in 2024 or last year. Last year. Okay. So this is also a very good article. Then coming to the therapeutic proteins, it's a very important tutorial part. So because there are some wet lab here, so major challenge is about we are talking about industrial protein. Now therapeutic proteins means the enzymes used for curing of many diseases. Okay, which everybody is talking about, including the growth factors, very costly. Okay, when you talk about these enzymes and therapeutics, everybody is very much afraid of the cost. Okay, why the cancer treatment cost is very high? Because of the costly medicines. So therapeutic proteins, they play a very important role, and among them growth factors, cytokines, monoclonal antibodies, and all other aspects are there. Work is also done. Okay. So when we talk about that, there is something which requires more critical thinking. So with this few slides, I will show you why we thought about it and how we addressed it. It's a case study, small case study. I am not presenting all the slides. All the slides are more than 100, okay? Because it will have wet lab data also and all those things, which I can show you sometimes else another day. But if you see this, we try to see the role of this enzyme pyruvate carboxylase, PC. Okay, so what is happening is that the PC is connecting glycolysis to TCA cycle in metabolic pathway of recombinant cells and generate energy in the form of ATP. Low enzyme activity leads to acidification of the medium due to accumulation of the high concentration of lactate. Lactate is a waste product. So what is happening because of this enzyme, low activity of this PC enzyme, okay, the activity of particular molecule is not coming up to high. So what is the target? What we can do as a biotechnology person or who are attending this course for drug discovery? So when we talk about this, lactate accumulation can be reduced after reading lot of literature, after reading lot of other papers. We understood that okay, we can do metabolic engineering, we can do some more work on the process, we can see more structural design, we can have structural studies, and we can do the process optimization and cell engineering to improve the lactate accumulation reduction. And if we reduce, the PC connector will be better. And if it is better, more connectivity, more enzyme will be available, or there will be some more activity of that enzyme for therapeutic yield. So what we did is that this is the strategy which we used, but it is a customary process. Means normal we use it. Okay. What we do is that we do, if we want to have some products, okay, like therapeutic protein or whatever product you get from fermentation, because all product which you are producing for the therapeutics or for the drug, it is all due to the fermentation and large scale production. So you do like this, okay? You do the purification, first you do the desired production from microorganism or co-clone, then you do fermentation, then you do the purification, and then you do the final product. Then market drugs and packet and sell. This is a normal process. But this process is not enough. So these days we have adopted this process, and this process is successful because we have completed our wet lab study. Now here this slide is having lot of idea. Okay, you do innovation in all steps. Innovation in all steps. How you do that? Clone development. You develop a better clone. You make a good clone. You try to make a high producing clone. Then your product yield will be better. You do improved fermentation. You have a small microreactors. You use the process scaling. You use the medium and feed optimization with new methods, AI based methods. You get a better product, more yield. You do innovation in purification. Simple purification, and you use the tanks, various fusion tanks. Those who are working in animal cell technology, they know that. You use different tanks. So if you do these innovations with advanced technology, you get cost effective production as well as high yield. If the cost is high, it can be reduced at subsequent step with better clones. So in clone development itself is not easy. You can do glycoengineering, you can select novel promoters, you can use the various fusion tags, okay, and these technologies are giving you better output when you talk about the therapeutic product. So conventional technology to advanced technology. Conventional technology, everybody does it. Advanced technology, less people do that. So this strategy we proposed, and we also adopted it in our research. So I will show you that one. This is the tutorial review article, Advanced Technology for Improved Expression of Recombinant Proteins in Bacteria, Perspective and Applications. This paper is highly cited paper. You see in Google, this was written by us, and the person who is there, Sanjeev Kumar Gupta, after completing PhD with me, he got the top rank. He is now Vice President of IPCA laboratory. Okay, so he joined as a researcher. He was a scientist there after this discovery, because the company made million of rupees, million of dollars with that type of technology, and they funded also for that. And now after you see this aspect, which is advanced technologies for improved expression of protein, if you are really having good expression, high yield, then your problem is sorted out. So this is the biggest challenge which is possible, and conventional technologies, they are evolving, but that they are not able to give a good production. So we say that it's a novel platform or microbial platform technology. So this technology we have developed in our lab in collaboration with the IPCA laboratory, and this technology will give you a very improved and high efficient production of the therapeutic proteins. So we have not targeted all the proteins. We have targeted MAB, that is monoclonal antibody, in general in PC engineering. So I will show you that. Before that, you see this. Okay, this is the big problem. Okay, and this problem need to be sorted out. Lactate and ammonia, both are accumulated in the cell, and they create lot of problem. You know how they are accumulated, and how to get rid of that is a big challenge. Okay, so cell viability is reduced, product quality is reduced, productivity is also reduced. So these are the common waste products which are accumulated, and Now how to do that and how we have done this. We did clone screening. We made different clones through various platforms and we generated 500 clones. Okay, 500 clones. Then we did the evaluation, single cell analysis of 47 clones. Then we did the top 10 clones, then top 5 clones, and then final clone. This all exercise takes 3 to 4 years or maybe more. If it is a university setup, industry setup, things are better, little better. They are in control of, okay, and that is open all the time. So if you talk about this, when you see the final clone, you can't do all this with all the clones, okay, and you don't, if you don't have 500 clones, you don't have much options. So you have to select maximum clones which you can have in your research lab. So these clones, and then evaluate, then try to see the copy number, how much copy number study. This is also very important. Then you select top 5 clones, and then one clone which I have written at clone 12. I will show you that. You do these studies, and if you are able to do only this one and you have not done, it is of no use, because if you don't have a clone purity, you don't have good metabolic profile, you don't have protein RNA expression profile, you don't have copy number and stability, which is very important. Okay, so you cannot go ahead with your therapeutic protein development or therapeutic protein production. Okay, so we go in clone processing. This is a flow chart for that. Please note it down. Then clone selection criteria, going one by one. Clone selection criteria should be based on cells growth behavior, metabolic profile, process feasibility, chirality and stability, expression and gene load. If you are working on clone, if you see here, I have made some green, okay? Green means all good clones. So we are having very few green cells, green clones, okay? All good clones which are having good profile. So we cannot do all the work, okay? We have to take only the person, the clones, the specific type of resources which are really paying you better in the later stage. Because if you don't have all green clones, then it means your selection profile was not put properly, and it is all costly affair. So you do all these aspects in clone selection, and if you get meritorious clone, you go ahead further. So in this study, though I have another 20-30 slides to show, I'm not showing all that. I'm showing a nutshell to you. The whole paper is available online, so you can read the paper. Whole paper is there. Okay. The PC engineered the pyruvate carboxylase enzyme which we are targeting. We engineered that enzyme, okay, using the cell engineering and other techniques, and we controlled this to cell line development, and we provided a better platform with the help of this technique, and the therapeutic protein, especially MAB, which was targeting, was improved. And when we did the metabolic engineering of the PC gene into the CHO cell lines, cell engineering one, okay, we also got improvement in the performance. And these are the data which is done in our lab. Higher cell density, we got 26 million cells per ml. Okay, we got culture longevity, that was two times more. We got lactate consumption, you see that this was there. Then glycosylation 2.5 fold increase. Then significant increase in MAB protein expression, that is 70% MAB. Then charge variant, we did the charge variant study. This was the first study. I will show you the comparative table. We didn't got good significant results, okay, but that is also results. So I am telling you. Then optimal PC gene 3-4 copy number, this is reported for the first time by our lab. Then we also used the first time codon optimized. So there were earlier studies, they didn't use the codon optimized aspects for cell line engineering and metabolic engineering together. So we did this study, and you see this is a comparative. I told you in the beginning, compare. What you are doing, compare. Okay, we are the best, we are the best. But see how other people are doing. Okay, so we compared where we are. Okay, so this is the present study, this is the other published study, these are the references. You see, we use this CHO cell line suspension. Other people, they use this type of cell lines. Then we did the codon optimized, but there was no report of the codon optimized earlier. So this is innovation which we did. We did the codon optimized yeast pyruvate carboxylase. Then PC gene copy number also on lactic acid metabolism first time reported. It was not reported earlier, and gene copy number was not available. Highest peak density 26 million. I showed you. That is the cells. How many cells are there? We got earlier there was a report of 14 million per ml. So we got 26 million. Okay. Then very high lactate consumption rate up to 4 folds compared to the control cells. Little high up to 25%. Then cell viability. This is another very important factor. We got cell viability on day 14 up to 94%. Means 94% of the cells were viable even on day 14. It means longevity is there. The reports were showing the cell viability 75% on day 12. So day wise also 14 and percentage wise also 94%. Then titer 76 at pool stage. So when we are generating the pool of cells, we got 70% improvement. And here the reduced titer was reported. So we have claimed that. Then galactosylation by 2.5 fold and mannosylation by 2 fold, and data was not available earlier. No significant impact on wire charge variant. So we did the study again to see, but we got the same results. Okay. So these are the highlights how you can plan, and if you really want to target therapeutic protein production, MAP production, it's a very good area. And though our lab right now is not focusing much on that, but if some of students they are interested, we can think about it, because still there is a lot of study which we can think about and we can plan. But we need to see whether, because we have to work on this multi-focus angle. It is not only one angle. You have to see that your product goes to the market. So when you connect with industry, okay, industry, academia, the research you are doing is a need based. So like the lab is asking, IFCA lab is asking, okay, this is the problem. So we need to understand that problem and address the problem. And once we address the problem, we get this study, they use the technology. So it is all win-win situation for both of us. And this type of a study is very important. And we published this study in PLOS 1, and it is all collaborations. You can see, and this lab is there in Mumbai. Whenever you visit, you can go. You can meet Sanjeev Gupta. He is there, and he will show you the high-class facilities there. Earlier also were there. Now he's vice president, so you can go and ask him what new things. But they are now focusing more on especially neurology. Okay, so earlier also they were focusing on growth factors and all that, and they are biosimilars. Okay, if you talk about biosimilars, I'm not talking here today, but biosimilars is also very good, and this study is published. So you can read this paper. Isme pura all protocol is there. Okay. Then this tutorial article, microbial platform technology for recombinant antibody fragment production, also very highly cited article. We published in critical reviews in microbiology. Okay, and these all journals, they hold a great impact, especially when you publish, when you read, when you cite, and then make notes out of it. Because if you do not read, you cannot understand the state of art. So this is very important. And here in this, we have also covered this Wacker's secretion technology for expression of the FAB, especially when you talk about the production in the fermentation crop. And also the Pepnet system and also the expression vectors is also analyzed here in this paper. And this is gene editing for cell engineering. It's also a very good tutorial paper. You can see those who are working on cell engineering aspect. Cell engineering for drug development, that is also there. So they can read this paper and try to know how the things should be worked out. And those who are working on multiplexing gene editing approach, they can also read this paper. Okay. And I am very happy to tell you that these papers are cited by high impact papers of science nature. Okay, I have a report. I am not able to show you here. Maybe tomorrow I will show you how many our papers which we have written in these emerging areas are cited by science nature patents, US patents. Okay, and this study, the analytics is given there, so you can see that from your idea others getting idea, and they are not getting only idea, they are executing the idea. Okay, so we generate idea but we are not able to consolidate, then again we have to rethink that, okay, how we can improve. So this is all about learning. Okay, and this is all the techniques. Okay, this paper complete tutorial paper, that's why I have written here. Sophisticated cloning, fermentation, purification for enhanced therapeutic protein production. Also available online. You Google it, you will get it. It is free. Okay, this and PLOS ONE, both are free. And this is Frontier in Pharmacology. And this paper will also give you the purification technology, which is very important. The major challenge is purification. Many students, they come and say that, okay, the purification is a big challenge. So this paper gives you all aspects of that. Okay. Then this is the paper where we have that lactate metabolism, the second part of it. And the story with that is that this PYC2 enzyme is a very crucial enzyme, and if you have this enzyme, especially when you are talking about MAB production, so you cannot forget the role of this enzyme. And when you use the cell culture improvement, you use all the cloning improvement, you target various type of specific technologies which I showed you in the previous slide, you can definitely get higher yield, higher production. And you know the cost of MAB. You know, I cannot tell you right now, you Google it, you will get it. Okay, everything is available in Google. Market research on growth factors, especially the medicines which are using these type of products. Okay, so there is a lot of demand. Okay, so now other future lines of research you can target, which is related to drug discovery. One is the probiotics and prebiotics. Okay, the first one is not much related to drug discovery, it is related to environment. Okay, but those who are working in environment also, they can see. And there is a lot of structural biology for environmental aspects also. So structural biology is everywhere. Okay, and if you know the structures, if you know many things, function also you know. So biocomputational tools in probiotics and prebiotics, this is also least worked area. Okay, how the probiotics are behaving, gut-brain axis. Okay, how the medicines, how the various type of drugs, okay, and how these gut microbiota modulation is capping. So these are aspects. Then metabolic models and microbial interaction. See, most of the diseases due to the microbial interactions. Positive interaction, negative interaction. So whatever interaction is happening, they will have some type of symptoms and then diseases. So you understand these aspects and try to see this aspect as well. Then further, if you try, you see this slide. I told you in the morning. For learning, for earning, for giving, for taking, and for joint understanding, everything lies in this map. We need to learn from many researchers. It is not always that one researcher, one time, two researchers, okay, three researchers. If you see, we are collaborating with these researchers sitting at here, okay, and it is all last 15 years. We have been collaborating with many people, okay, in different area. Like our lab works on tools developments. When you talk about techniques and tools, we are the person who are interested in tools and techniques development. If somebody tells, okay, I want to develop this tool, we are the first to help that particular person. So if you see many areas, like if you want to work on enzymes, Kavra Bayasi Utaka and Professor Suren Singh is here, okay, especially the industrial enzyme. If you want to talk about the probiotics, you have a person here, that is the Jingmin Sun. He is working there in US. Okay. Then if you talk about the other parts of this biofuel and all that, you have the Jacob Leffers, then Sangeeta Neri, if you want to work on tissue engineering, because we also wrote a very good tutorial review article, Advanced Functional Material, BioCPS, Bio Cyber Physical System, that is also part of drug discovery. Okay, cyber physical system, new area. Okay, so there we are collaborating with Henning and Magali, Germany. Okay, they both are doctors. Okay, MD. If you Google, you will get. So Magali and Henry, we wrote a very good paper together with one of my previous student, Prasoon. He is now assistant professor in NIT Rourkela. So we wrote that part, and tissue engineering is one of the very important areas, and especially getting that. Then he is the big guy, Omri Koren from Israel, small country here, okay, and he is a leader in probiotics and prebiotics, microbiome engineering, Omri Koren. Okay, so we have to learn from each other, collaborate from each other, and then this is the GIAN. Okay, what we are talking is the GIAN, and GIAN is coming from various aspects. And Professor Joseph Chang is there, then Amit Rai is there. Amit Rai, I think you interacted earlier. He is right now in US, I think he's in US. Then Professor Utaka is there, then Manoj Kirao, Professor Patel, then so many people. Okay, so thanks to them for training me and helping me and training my students and giving their knowledge, because giving knowledge is also a big thing. And this is present our lab, so because I wanted to thank all my lab students who are working hard. So this is our present lab. Every student is involved. These are two MSc students here from this batch. Previous batch I have written and made photo of few of them, but not other batches. Okay, so we are a small group working on various areas, and our lab is techniques and tool based. So if you are interested, you are always welcome. And Srabni is there, you can see Namrata is there, and Nivas was sitting, and these are our lab alumni. If you see, the whole work which I have presented you has been done by other alumni also, so I should acknowledge all of them. So if you see Twinkle, she worked on binocular development, and right now she got postdoc in this South Dakota University, US. Then Monica, she is an independent now working. She has not she was working with probiotics and prebiotics. Dinesh working with Bionest BU. Then Sunita Verma is in AIMS right now. She has a paper on computational vaccine design. Okay, and she worked with in collaboration with Professor Yogen Singh at BU. She is working very well in especially the vaccine design. So that is a good area, drug development and this adjuvant and all that. Then Ishu is working as assistant professor in a private university. She worked on probiotics. Then Babita, she is also working in some college. Then Shweta, she is working as a postdoc. She works on microbial remediation. And Mandeep is sitting here. Okay, he is assistant professor in Ansraj College. He specializes in the enzymes, especially industrial enzymes. And then these are the trainees, because we need to learn. So we have trainees from different backgrounds. So these were trainees when they came in my lab. Now they are not trainees. Okay, they are all well placed. Rajat is a postdoc fellow in Australia, senior postdoc fellow, and he is having a startup. In the morning I was telling, he is looking for collaborator from India, student and researcher who can provide him the information about personalized meds. So they have a big startup. Okay. And Arun is now not in research, but he was very good in protein and enzyme engineering. Ashwini MTech in he is also MTech in mechanical engineering, not biology. Okay, mechanical engineering. He came in the lab. We learned mechanical engineering tools from him, and then we also gave him few knowledge. And now he is a very good biotechnology engineer. I say bioengineer. Okay. Then Ashwini, he trained our students for optimization tools, AI based. Okay. Dinesh also MTech, and he artificial intelligence and bioprocess. So these are the other lab alumni. Sanjeev, I already told you. So he did PhD and he is now in IPCA, vice president. Jahangir is in forensic science laboratory. Rameshwar is in South Korea. Chiranjeev is an assistant professor at Gurukul Kangadi. Shripal Vijayvargi is the first person. He was my colleague. He is now professor. Okay, so he was the first person whom I learned computational biology in 2009. Okay, he was in computer science department as assistant professor. I was assistant professor at biotechnology department. He was not having PhD, so he came to me and told that you already PhD, so you should tell me how I can learn research and all those things. I told, if you teach me computational biology or computer, so he told, biology I don't know, computer I can teach you, or computational tools. So we collaborated, then I guided him for PhD, okay, and then he's there. Then Puneet is protein engineering and bioinformatics. Recently he visited. He's a senior postdoc in South Korea. Then Vishal is in also South Korea. 3 of them are in South Korea. Okay. 4th one is going now. Okay. Smriti is in MIT, senior assistant professor. Ruby Mahak is in US. I think she has started a company, small company, because she worked as a postdoc. Then she started a company. Then Vishal in IIT Indore completed. This is our lab group photo. This is an old photo, and I don't think whether you can recognize anybody because nobody is here. They are all well settled. Okay. So bigger picture. Okay. So I think this is, again, I acknowledge all of these because, and thank you. So I think we are on time. Another 5-10 minutes are there, so we can have a discussion and question answer. If you have, please. I tried to summarize the part of therapeutic protein. If you want more details, I can cover in the next lecture because I have many slides. Okay, I can also show you the specific batch fermentations, what we have done, how the map production was done, how we took. I think this is important. I will show you those techniques because those are all techniques. Okay, and these techniques are done to achieve excellent map production, and the map production profile increase was because of using those techniques. I think she's sleeping. Headache, medicine, fever medicine chahiye, drug. Medicine is there. You can ask. You go to the office, you will get it. Okay. If you want, if you are having headache because of lot of lectures, it is not easy to join this program. 1 week, lectures. But anyway, thanks to you, and your presence is marvelous. So you may have any questions, please ask, because we have another 10 minutes left. Any questions? So if you don't have any question, I can ask few questions. I can ask few small questions. From after discussion, this post-lunch session, what we learn basically, the major summary of the research, what we are doing, what techniques we have read today, what we learned is that, can you anybody tell in 1 line or 2 lines? Or bullet points, 1, 2, 3, 4, that will be better to summarize with that. Because so many things we discussed, so many things I don't know whether you noted it down or not. And something which you have not noted down, that is more important. Because it may skip later on and you may not be able to get that idea that what was there. The one thing which you take it from today onwards, if you really want to be in research, if you just do your MSc, do some other work, it's fine. But if you want to be in research, plan the techniques part in advance. What techniques you are going to use in your research. Very important. In MSc program, those who are in MSc, we have 6 months devoted for techniques. Okay, and most of the time, if you are knowing which techniques you need to use in research and what technique you will like means compatibility, facility, okay, and feasibility, okay. So if you are planning your research for the PhD students and master's students, techniques part is very important. You plan it in written way, not only in your mind. You plan it properly and see the techniques which you are using in your research, really they are advanced. If they are not advanced technique, the publication potential will be not high, because the routine techniques, routine experimentations, routine analysis is not having good impact, okay, because the things are already known, okay. So if I tell you yeast cell, I will show you the yeast cell, okay, you will know that yeast ka pata hai kya hota hai. I show enzyme production, you know enzyme production. Okay, in the same way, if you do some type of immunogenic response, it is people they know it. So you plan it based on your requirement, the techniques part. This is the first bullet point, okay, in list. Second bullet point is about the planning, TOC. If you are really interested in a good research, plan a TOC. इस step के बाद ये और what will be the alternate plan? What will be your alternate plan if you fail, okay? But don't always think that I will fail, okay? If you fail, or if your experiment fail, or if for any reason you cannot carry out that, what is your alternate plan available? If you have that alternate plan available, I think you will be more confident, okay? That okay, if it doesn't click, we will go to the next one. So first plan, plan A and plan B. So plan B should be there with your records book. Okay, you maybe you share with your mentor, with your colleagues or not, that is up to you. But you should have a plan B, and it is always good to share with your mentor, because your plan B feasibility has to be cross-verified, checked by the mentors. The third bullet point is comparative analysis. Whatever research you are doing, do a comparative analysis whenever you get time. That whatever you are doing, really it is the new things which you are exploring, or you are understanding the concepts only. Okay, understanding concept is not a problem. Okay, it's okay, learning process. Okay, but if you want to really come up with a difference, then you have to have a comparative analysis. With same molecule, same enzyme, same type of drug development, many people are working on diabetic, anti-diabetic drugs, cancer drugs. Many people, not only I and you are working, many people are working. So you have to see that what specific things they are carrying out and how they are making difference. Okay, whatever papers are published in high impact journals, you read it, see what is the basic thing, because sometimes they look very simple. Okay, but what is the real technology in that paper? Okay, so this is the third bullet point about the comparative overview. The fourth bullet point, you have to tell me. What is the fourth bullet point? The last slide I presented you. Exploring new area and collaborations. Exploring new area and collaborations. That is very important. Don't be afraid of collaboration. Okay, I think that nobody can steal your data, nobody will take your data, nobody will take your idea. Idea बहुत सारे हैं, सबके पास idea है, काम करने वाले कम, ठीक है? So exploring research collaborations and also the ideation, that is very important. So this is the fourth bullet point. From this session, I think if you focus on 1 and 2, you will be successful. I am not telling all you can focus. 1 or 2, you will be successful, I am telling you. Because when you make TOC, definitely you will read the papers and you will try to collaborate with people. So they are all interconnected. And if you think that you can do everything, this is impossible, I am telling you. You can't do everything together. I cannot do everything together. I am dependent on many other researchers who are working in area, and I am very open to discuss with them, to share with them. And sometimes you cannot be expert of everything. Okay, at least you should know that fact. Sometimes you think that okay, I will be able to complete all myself. No, you should focus real learning strength. Okay, and I think probably after a few lectures by Professor Tong and other few sessions, you will realize that why collaboration is very important. Can you show a single paper of either Professor Tong or mine, okay, with one or two authors? Very few papers, few papers you will get, but most papers you have 3 authors, 4 authors, and with different institutes. He is also collaborating with various institutes. And that's why he was also telling me that for high impact research, we need to understand the power of collaboration. Okay, in these days, people they say collaboration को short में collab बोलते हैं, ठीक है? So people they say collab. So this is collab, and if you do collab, you have collage. Okay, so collage of research, collage of good quality research with help of collab. So finally, I think this is now time is over. So we close this session, and if you have any queries, papers whatever I have suggested you, please note it down. Any of the student, MSc student, they can note it down and take those papers, okay, or the paper soft copy is available. The only thing is that we cannot circulate it, mass SS circulation, and it's uploading on ResearchGate or something, okay, because the publishers, they say that, okay, this is embargo period, you don't do that, okay, so it is all copyright policy that papers cannot be circulated freely if it is protected by some license or something. So I don't understand that, it's by the publisher's policy, but the papers are for academics, and those papers which are open access, you can definitely use that paper, okay, and keep scrolling new ideas in research, and I wish that all of you, all 60 people who are registered for the program, they will be a good researcher in coming years, and they will come up with good papers, and I wish to listen them sitting there, okay, with their discoveries in the coming years. Okay. So with this, I think we close the session. And any questions? Last questions. 1 minute is left. If you have any questions, we meet tomorrow at 10 again. Okay, and at 12 we have arranged some snacks for you because I feel that your energy is required. So we have arranged from tomorrow some snacks at 12 break. Okay, some sandwich I had told him to give. Okay, so probably they will, and on the Friday we have a lunch. Okay, so that is there, full lunch.